Hello dear friends, welcome back again to another video from my YouTube channel. Dear friends, in our previous video, we have studied about the gametogenesis process. Gametogenesis are of two types because there are two types of gametes in the human beings, male gamete and the female gamete. Male gamete is the sperm and the female gamete is the ovum. The process of formation of male gamete, that is sperm, it is called as spermatogenesis. Whereas the process of formation of egg, it is called as, or ovum, it is called as oogenesis process. In this video, we are going to understand the spermatogenesis process in detail. How the sperms are getting formed inside the seminiferous tubules of the testis. That we are going to study in this particular video, my dear friends. This concept, it is very important for both board as well as for competitive exams. Why? Because from this concept, many questions have been asked in the previous year question papers or previous year competitive exams like AMU, NEET, AIPMT, even the JEE, Gujarat CET, and this question was asked in the Karnataka board examination, second year PUC board examination for 5 marks in 2019 and in 2014. So this concept, it is very complicated process. I made it very simple for my dear friends. Okay, let us begin the स्पर्मैटोजेनेसिस प्रोसेस. अब ये वीडियो में हम स्पर्म्स किस तरह से बन रहे और कहां बन रहे पूरा डिटेल में समझने की कोशिश करेंगे. लेट अस स्टार्ट. If you recall the definition of the spermatogenesis, spermatogenesis means the process of formation of sperms inside the seminiferous tubules of the testis. What is the location? Let us understand the location first. In case of human male, we have a pair of testes located in the scrotal sac. A pair of testes. These testes, they show certain compartments in them. These compartments are nothing but the testicular lobules. Approximately 200 to 250 such compartments are there in the testis, human testis, in each testis. Now, inside these testicular lobules, there are some highly coiled tubular structures are present. These tubes are nothing but the seminiferous tubules, my dear friends. Now, let us take out this seminiferous tubule for our better understanding. Seminiferous tubule is a highly coiled structure and it is a tubular structure, highly coiled tubular condensed structure it is. If we take the transfer section, of this seminiferous tubule, how it appears? It appears like this. If you take the transfer section of a seminiferous tubule, if you take any transfer section of any tube-like structure, this kind of sectional view we are going to observe, my dear friends. Here, this seminiferous tubule, it consists of two types of cells. Some of the cells are conical cells. We talked about these cells in our previous videos while discussing the sectional view of a testis. They are the conical cells. They are nothing but the Sertoli cells. Whereas one more kind of cells we have in the seminiferous tubule of the testis, and they are cube-like, like this. They are the flat cube-like cells. These cells, they are called as spermatogonia cells. There are two types of cells present in the seminiferous tubules of a testis. One is Sertoli cell and the other one is spermatogonia cells. Now, let us take only this much portion for our understanding. Let us take only this much part to understand the process. This is the spermatogonia cell. Now, if I enlarge this, how it appears? It appears like this. Here we'll have some pyramid shaped cells. These pyramid shaped cells are nothing but the Sertoli cells. They are the Sertoli cells. Whereas one more kind of cells that we have, they are the spermatogonia cells. Spermatogonia. They are the spermato. Let us write here. They are the spermatogonia. Spermatogonia. Now, to understand the process of sperm formation in case of male, the scientist has divided the entire process of spermatogenesis in 3 different phases, in 3 different steps. Let us talk about the steps first. The first step of the spermatogenesis, it is multiplication phase. Multiplication phase. Then the second step is growth phase. Growth phase. And the third step is maturation phase. Maturation phase. They are the 3 distinct, they are the 3 very important steps through which the sperm production takes place inside the seminiferous tubules of the testis. We'll understand what is multiplication phase, what is growth phase, what is maturity phase little later. Now, there are 2 types of cells in the seminiferous tubule of a testis. One is Sertoli cell, other one is spermatogonia cell. Let us focus on this spermatogonia cell. Let us focus on this spermatogonia cell. This spermatogonia cell, it starts dividing by a mitosis. It starts dividing by a mitosis process. How is this spermatogonia cell, my dear friends? Any cell which is related to the body, any somatic cell, it is always diploid. It is always diploid. All the cells are diploid. All the cells are diploid. Even the Sertoli cells, they are also diploid. All the cells which are there in our body, they are diploid in case of human beings, except the gametes. Sperms are haploid in male, whereas ovums are haploid in case of females. Apart from sperms and the ovum, all the cells which are present in the human body, they are all the diploid. That means definitely they consist of 46 chromosomes, my dear friends. Now this cell divides by mitosis. As a result of that, 2 cells are formed. 2 cells are formed. This division, it is mitosis. It is mitosis. We know mitosis is an equational division, where the chromosome numbers, they remain equal even in the daughter cells also. That means if this is a diploid cell, definitely the daughter cells, they are also diploid, because it is a mitotic division, my dear friends. Now these 2 cells, they again divide by the same mitosis process. They again divide by the mitosis. As a result of that, we'll get a 4 cells. Here, this division is also mitosis. It is also mitosis. It is also mitosis means definitely they are also diploid cells. Diploid cells, let us keep a track of the ploidy chromosome number so that we can understand the concept in a better way. Now, these 4 cells in future, they give rise to 8 cells. From 8, 16, 16 to 32, 32 to 64, like that. The number of the cells, they keep on increasing by mitotic divisions. अब मैं तो पूरे 46 सेल्स यहाँ पे बोर्ड पे नहीं निकाल सकता, 8 सेल्स बोर्ड पे नहीं निकाल सकता, 16 सेल्स बोर्ड पे नहीं निकाल सकता, इसलिए मैं 4 सेल तक अब इसको महदूद रख रहा हूं, मैं 4 सेल्स को जिसको मैं स्टॉप कर रहा हूं. बट द सेल नंबर, it keep on increasing by the mitosis. As a result of that, many cells are formed. Many cells are formed. All these cells, they are still the spermatogonia cells. They're still called as spermatogonia cells, my dear friends. Now, half of the cells out of 4, ಇದರಲ್ಲಿ ಕೇವಲ 4 ಸೆಲ್ಸ್ಗಳು ಹುಟ್ಟಂಗಿಲ್ಲರಿ, ಇಂಥ ಬಹಳಷ್ಟು ಸಾವಿರಾರು thousands of cells are formed. Out of thousands of the cells, half of the cells, they are called as type A spermatogonia. Type A spermatogonia. Half of the cells, 50% of the cells, they are called as type A spermatogonia, and remaining half of the cells, they are called as type B. Type B spermatogonia. Spermatogonia. Suppose 1000 cells are formed. 1000 ಸೆಲ್ಸ್ಗಳು ಉಟ್ಕೊಂಡು ಅಂತ ಹೇಳಿ ತಿಳ್ಕೊಳ್ರಿ. 1000 ಸೆಲ್ಸ್ಗಳಲ್ಲಿ 500 ಸೆಲ್ಸ್ಗಳನ್ನು ನಾವು ಟೈಪ್ ಎ ಸ್ಪರ್ಮ್ಯಾಟೋಗೋನಿಯಾ ಅಂತ ಹೇಳಿ ಕರಿತಾ ಇದ್ದೀವಿ. ರಿಮೈನಿಂಗ್ 500 cells, they are called as type B, type of spermatogonia ಅಂತ ಹೇಳಿ ಕರಿತಾ ಇದ್ವಿ. Type A cells, they continuously divide by mitosis. That means they remain as a stem cell. Their main job is, half of the cells main job is to divide continuously by mitosis. Whereas type B spermatogonia cell, they enter into the next phase of the spermatogenesis process. They enter into the next step of the spermatogenesis process. What is the next step? Let us keep a track of this cell only. Here 2 cells are formed, but I am tracking only 1 cell for my convenience, for your convenience also. Let us keep a track of this cell. This cell, what it will do, it enlarges in its size. Half of the cells, they become type B and they enter into the next phase of the spermatogenesis, whereas half of the cells, they remain as a stem cell and they keep on dividing by mitotic process. They are multiplying. Up to this, not here, up to this stage, it is called as multiplication phase, the first phase of the spermatogenesis, where the number of the cells, number of the spermatogonia, they are continuously increasing by mitosis. Mitosis ಮಾಡಿ ತಮ್ಮ ಸೆಲ್ಸ್ಗಳ, ತಮ್ಮ ಜೀವಕೋಶಗಳ ಸಂಖ್ಯೆಯನ್ನು ಹೆಚ್ಚಿಸ್ಕೋತಾನೆ ಹೋಗ್ತಾ ಇದಾವೆ ಸ್ಪರ್ಮ್ಯಾಟೋಗೋನಿಯಾ ಸೆಲ್ಸ್. ಆದರೆ ಅರ್ಧ ಸೆಲ್ಸ್ ಏನಾದವಪ್ಪ ಅಂತಂದ್ರೆ ಟೈಪ್ ಬಿ ಆಗಿ ನೆಕ್ಸ್ಟ್ ಫೇಸ್ ಅಲ್ಲಿ ಎಂಟರ್ ಆದವು. ಈ ನೆಕ್ಸ್ಟ್ ಫೇಸ್ ಅಲ್ಲಿ ಎಂಟರ್ ಆದ ತಕ್ಷಣ all the type B spermatogonia cells, they increase in the size by getting nourishment from the Sertoli cell. If you recall the function of the Sertoli cell, the very, very important point for the competitive exams, my dear friends. What is the function of a Sertoli cell? Sertoli cell's main job is to provide the nourishment to the developing sperm, to give a nutritive material throughout the spermatogenesis process. That is the main job of the Sertoli cell, my dear friends. Now, after getting the nourishment from the Sertoli cell, the size of the cell get increased. Now, when the size of the cell increase, it is no more called as a spermatogonia, it is called as primary spermatocyte. It is called as primary spermatocyte. Here, if I ask you a question, the very tricky question now. Here, this cell, whether it is haploid or diploid, it remains diploid, my dear friends. Why? Because there is no reductional division takes place, only the size of the cell get increased. अगर मेरा नाम आसिफ सर है, अगर मैं मोटा हो गया तो मुझे क्या बोलेंगे तुम? आसिफ सर बोला छोड़ के दूसरा क्या तो भी नाम दे सकते क्या? नहीं, मुझे मोटे आसिफ सर बोलेंगे बस. अब इन्हें मोटा आसिफ सर बुलाने से बेहतर है, it is called as primary spermatocyte. It has given with a different name, primary spermatocyte. It remain diploid, only the size is increased, my dear friends. Now, this primary spermatocyte, it undergoes meiosis. Now, it undergoes the meiosis. Let us write with a different marker. It undergoes meiosis. We know meiosis is a reductional division where the chromosome numbers reduce to half, my dear friends. And the meiosis is completed in 2 different steps, meiosis 1 and meiosis 2. First, there is a meiosis 1. Meiosis 1. As a result of that, there are two cells are formed. Two cells are formed. If you recall your first year knowledge, cell division and cell cycle chapter, meiosis 1, it is the reductional division. Meiosis 1 is the reductional division. Whereas meiosis 2 is just like a mitosis. Meiosis 2 is an equational division. Meiosis 1 is a reductional division. If this is a reductional division, can you imagine, can you give me an answer whether these two cells, they remain haploid or diploid? Yes, my dear friends, they become haploid. From diploid, now they become haploid due to the meiosis 1, which is reductional. Now, as a result of that, these two cells, they again divide and they complete the meiosis 2 process. Meiosis 1 यहाँ पे हुआ बोले तो अब ये आगे चल को meiosis 2 करने वाली है दोनों cells. Now here it is a meiosis 2. Meiosis 2nd. As I told earlier, meiosis 2 is just like a mitosis. It is equational division. Meiosis 2 is a equational division. अब ये meiosis 2 equational division है बोले तो अब इससे दो जो cells बन रहे हैं, these cells, whether they remain diploid or the chromosome number reduced to half now. No, my dear friends, here the cells, they remain haploid. They remain haploid because meiosis 2 is a equational division. Now, we forgot to mention the name here. These two cells, after completing the meiosis 1, they are no more called as primary spermatocyte. Now they are referred with a different name. They are secondary. They are secondary spermatocytes. They are secondary spermatocytes. Secondary spermatocytes, they undergo meiosis 2, which is equational. As a result of that, 4 cells are formed. All these 4 cells, they are called as spermatids. Spermatids. Remember the names. They are very, very important, my dear friends, for competitive exams. Here, these cells, they are all called as spermatogonia. From spermatogonia, primary spermatocytes are formed. From primary spermatocytes, secondary spermatocytes are formed. From secondary spermatocytes, spermatids are formed. Spermatids are formed. Now, I am taking all these spermatids here. I am drawing the same structures. 4 spermatids are formed. And all these 4 spermatids are haploid. They are all haploid. What are they? They are the spermatids. They are the spermatids. Now listen very carefully, my dear friends. These 4 haploid spermatids, they undergo certain modifications. They undergo certain changes. They undergo certain alterations in them. What are the alterations? Let us write them. Let us write the first alteration, first modification. Let us write here modifications. Modifications in spermatids. Modifications in the spermatids. The first modification that takes place in the spermatid is the condensation. Condensation of nucleus. The nucleus, here we have the nucleus. This nucleus, it becomes condensed. It starts becoming condensed. Then the second one is nucleus become conical. Nucleus becomes conical shape structure. Then the third thing, is loss of cytoplasma. All the 4 cells, they lost their cytoplasma, their nucleus become condensed, their nucleus become conical in the shape, and mitochondria of all these 4 cells, mitochondria arranged spirally. Mitochondria, it starts getting arranged spirally inside these cells. Then the fifth one is Golgi complex. Golgi body converted into a very important structure, that is acrosome. Golgi body get converted into acrosome, and finally tail formation takes place. They are the changes occurred in the spermatids. They are the six different changes, very important changes occurred in all the 4 spermatids. As a result of that, these cells, they attain a proper shape, they attain a specific shape that exactly resembles like the human sperm. Now they become the sperm. Sperms. They are called as sperms or spermatozoa. They are called as spermatozoa. From spermatids, spermatozoas are formed by doing these alterations, by doing these modifications. This step is repeatedly asked in the competitive exams, my dear friends. From spermatids to sperm, this journey is given with a proper name, with a specific name. This process is given with a proper term. What is the term? It is spermiogenesis. Spermiogenesis. Very, very, very important term, spermiogenesis. What is spermiogenesis? Conversion of spermatids into mature sperms. Or you can also call formation of a mature sperms by the spermatids. It is called as spermiogenesis. Spermatid ಇದ್ದಿದ್ದು sperms ಆಗುವಂಥ ಪ್ರಕ್ರಿಯೆಗೆ ನಾವು spermiogenesis ಅಂತ ಹೇಳಿ ಕರಿತೀವಿ. Let us write the definition. The process of formation of mature sperms, from spermatids. Spermatids से sperm बनने वाले process को हम क्या बोल रहे हैं? Spermiogenesis. Whereas spermatogenesis means this entire process. Entire process is called as spermatogenesis, whereas only this much process it is called as spermiogenesis. After the sperms they become mature, after the maturity of the sperms, now they are all called as mature sperms. Once the sperms they become mature, they get released out from the Sertoli cell. They still embedded in the Sertoli cell to get the nourishment. What is the main job of the Sertoli cell? To provide the nourishment. They are still inside the Sertoli cells. After becoming mature, they get released out from the Sertoli cell. Mature sperms released out from the Sertoli cells. ನೀವು ಕೇಳಬಹುದು ಪ್ರಶ್ನೆ, ಸರ್ ಸರ್ಟೋಲಿ ಸೆಲ್ಸ್ ನಿಂದ ಯಾಕೆ ರಿಲೀಸ್ ಆಗ್ತಾವ್ರಿ? ಸರ್ಟೋಲಿ ಸೆಲ್ಸ್ ಒಳಗಡೆ ಅವು ಎಂಬೆಡೆಡ್ ಆಗಿರ್ತಾವೆ, ಸರ್ಟೋಲಿ ಸೆಲ್ಸ್ ಒಳಗಡೆ ಅವು ಸಿಕ್ಕೊಂಡಿರ್ತಾವೆ. ಯಾಕಪ್ಪ ಅಂತಂದ್ರೆ ಸರ್ಟೋಲಿ ಸೆಲ್ಸ್ ನಿಂದನೇ ಅದಕ್ಕೆ ನರಿಶ್ಮೆಂಟ್ ಸಿಗ್ತಾ ಇರುತ್ತೆ. After getting the complete nourishment, they get released out from the Sertoli cells. This process, it is called as spermiation. Spermiation. What is spermiation? Release of mature sperms from the Sertoli cells. But in your NCERT, the statement is given release of mature sperms from the seminiferous tubule. But I am not satisfied with that statement of NCERT. NCERT statement ನಾನು ತಪ್ಪು ಅಂತ ಹೇಳ್ತಾ ಇಲ್ಲ, ಆದರೆ ಆ ಸ್ಟೇಟ್ಮೆಂಟ್ ಅಷ್ಟೊಂದು ತೃಪ್ತಿಕರವಾಗಿಲ್ಲ, ಅಷ್ಟೊಂದು ನಿರ್ದಿಷ್ಟವಾಗಿ ಅವರು ಹೇಳಿಲ್ಲ ಅಲ್ಲಿ. ಬಟ್ the exact specific answer is the release of mature sperms from the Sertoli cells, not from the seminiferous tubule, my dear friends. Release of mature sperms from the Sertoli cell, it is called as spermiation, whereas the formation of mature sperms from the spermatids, it is called as spermiogenesis, and the entire process of formation of the sperm, it is called as spermatogenesis. Three very important definitions, and they are very, very confusing. Spermatogenesis, spermiogenesis, spermiation. They are three very important terms. What is spermatogenesis? The process of formation of sperms inside the seminiferous tubule of the testis. It is called as spermatogenesis. What is the spermiation? Release of mature sperms from the seminiferous tubules. Release of mature sperms from the Sertoli cell. It is called as spermiation. What is the spermiogenesis? It is a process of formation of mature sperms from the spermatids. Don't get confused between these three definitions. They are very confusing and they are very, very important for competitive exams. Now, this process to complete, approximately 74 days are required. 74 days. To complete one spermatogenesis process, approximately 74 days are required. So, it is an energy consuming process. From where this entire process is getting energized? It is getting energized from the Sertoli cells. I hope this concept is very, very clear to you people from many a times in the competitive exams, my dear friends. What is the correct sequence of spermatogenesis process? Correct sequence ಅಲ್ಲಿ ಹೇಳ್ರಿ ಅಂತ ಹೇಳ್ತಾರೆ. From spermatogonia, primary spermatocytes are formed. From primary spermatocytes, secondary spermatocytes are formed. From secondary spermatocytes, spermatids are formed. From spermatids, sperms are formed. This is the correct sequence of spermatogenesis process. I hope you people have got an idea about the entire process. Now, we forgot to mention these steps. Here, up to this, it is called as multiplication phase. Here the cell increases in its size. It is called as growth phase. Now from this up to this complete process, it is called as maturation phase. They are the three very important phases. First phase is multiplication phase, where only the cell number is getting increased by the mitosis process. In the second phase, growth phase, the size of the cells, they get increased after getting the nourishment from the Sertoli cells. And the third phase is maturation phase. And this maturation phase, it always shows meiosis process. Meiosis. Whereas multiplication, it shows mitosis. Remember, it is also very, very important for competitive exams. I hope you people have got an idea about the entire process of spermatogenesis. If you have any queries, any doubts, please ask me in the comment box. And if you like this video, please hit the like button and don't forget to subscribe to my YouTube channel for your future videos. Thank you so much.